Okay, so we covered section 14.2, talking about going from RNA to proteins, and I'm going to skip section 3 and move on to mutations and describe some different types of mutations and how they affect genes. So the definition of mutation is literally any change in an organism's DNA, and there are a lot of different types of mutations. So one type of mutation that you should be aware of for gene mutations is a point mutation where, sorry, my chair is squeaky. Gene mutation is where we have one nucleotide that's substituted for another. Okay, so we call this a point mutation. And there are different types of point mutation. One that's silent, which means it codes for the same amino acid because the code is redundant. There are more than one codons that can code for the same amino acid. So if it codes for the same amino acid, even though you had a base letter change, it's still good. Nonsense might, means that the change or the mutation has resulted in a premature stop codon. So let's just look at a mutation here where we're going to delete a letter, which is our next one. So we have frameshift mutation, insertion or deletion of a nucleotide. So if I take out a letter here, I have a sentence that made sense. The cat ate the rat. Okay. You know what? I'm going to back up. Sorry. Let's look at point mutation. So if I change the letter from the cat ate the rat, that sentence makes sense. It's 3 letters. Each 3 letters codes for a codon. If I click on, C and change the C to a B, the bat ate the rat. Okay, now I have a point mutation where something is substituted for another, and it may not fold correctly. If I delete a letter, get rid of the E, and then everything shifts over because the code is a triplet. So now I have thick etta te her at, and it doesn't really make any sense anymore. So the thing about frameshift mutations, okay, if it occurs very early in the mRNA or the DNA gene, it's pretty fatal. If it occurs at the end, it still might work, but it may not. Okay, so here's before the mutations where it's normal. We get methionine, arginine, threonine, and phenylalanine. I think that's what that one is. If we have an insertion, we insert a letter. Now I'll take a look at my amino acid sequence. They're completely different. Or if I take out a letter, okay, completely different. Some more point mutations. So I have in the notes nonsense, where ATC, if I change that letter, this is normal, TTC, and it turns into ATC. Now I have a premature stop codon. Here's a silent one, TTC, okay? All of a sudden the C becomes a T. Now take a look what happens to the mRNA level, AAG, AAA, and it still codes for lysine. So no change in the protein. All right, so I think this is just another diagram in your book saying that you can substitute letters, and it might just change one amino acid, and maybe that amino acid, it's still going to fold correctly because different amino acids have different charges. So let's just say arginine. Actually, I'm going to look up arginine. Arginine charge. So looks like it's a +1, and all of a sudden we changed it to histidine. That's also a +1. So it may fold correctly, okay? And then we already talked about insertion and deletion. Another type of mutations. Okay, so that was all gene mutations found on a gene. Another type of mutation is a chromosomal mutation, where it affects a large part of the chromosome. Lights are going dark here. So there are 4 different types of chromosomal mutations. Deletion, where you lose all or part of a chromosome. Okay, depicted here. So I just have the sequence A, B, C, D, E, F, and all of a sudden we lose B. Duplication produces an extra copy of all or part of the chromosome. So I have A, B, C, D, E, F, and all of a sudden it goes A, B, B, C, D, E, F. Inversion means that the chromosome breaks and reverses directions. So A, B, C, D, E, F becomes A, C, B, D, E, F. And then we have translocation, where part of one chromosome breaks off and attaches to another, and this is very common in cases of leukemia. So you can see that I have GH from some other chromosome that I'm not aware of that got broken off, and it's going to insert itself to this chromosome. So now I have AB, GH, C, D, E, F. Now, the effects of a mutation can be harmful, can be helpful, or maybe not have an impact at all. But phenotype could be impacted. So if it's a chromosomal mutation, it does affect a lot of genes. So you're going to see a big effect on the individual. Gene mutations tend to be minor, but they can be major depending on where it's at. If you remember in 7th grade, I showed you Tay-Sachs disease, where one letter change really messed up a protein that breaks down fat in the brain. Well, now that the protein can't break down fat builds up in the brain, killing critical brain cells, and the child goes blind and usually dies under the age of 5. Okay, so they can be major. Mutation in non-codon region can also have an effect, and then the substitutions could code for the same amino acid and as a result have no effect. So, case study of sickle cell anemia. So normal red blood cells kind of look like tiny little donuts, and this is what normal hemoglobin is made up of. Each one of these is a big macro or big polypeptide subunit that bonds together to form this massive protein complex, aka red blood cells. And sickle cell anemia is a disorder where one base change from GAG to GTG causes the normal hemoglobin, this nice little pocket area where it can hold oxygen, to change to sickle shape. So they form long, inflexible chains. And yes, that is a disadvantage in a lot of places of the world. However, in Africa, where malaria is an epidemic, it actually has an advantage. So let's just take a look at where malaria affects in the world, Africa and Asia. Okay, but if you take a look at Africa, this bottom slide is showing the frequency for those that carry the sickle cell anemia. So what is sickle cell? Sickle cell anemia, your red blood cells are sickled shape. But malaria, okay, is caused by a protist in the saliva of mosquitoes, okay? And so the protist lives in the saliva, so when a mosquito lands on an individual, it releases sporozoites into the bloodstream, and they target the liver cells, where they get released and they spread out and they infect red blood cells. And then they bust out and destroy that red blood cell, and then might get picked up by another mosquito or infect more red blood cells, and it's not good for that individual. But the advantage for sickle cell is that because it's sickle shaped and it's inflexible, the plasmodium actually kind of has a hard time burrowing through the structure. And
this is Lumen, the first device.
I have a video that I was going to show you, and so it's actually an advantage. So I have a video.
It's for hacking your metabolism. With just one breath, Lumen tells you what you're currently burning for energy.
Actually, if I remember right, you know what? I'm just going to post the link because this is kind of one of those weird videos that I found. And yeah, sorry. I kind of like to dabble on that. Okay, the impact on the offspring. Mutations that occur in your body cells only affect that organism. So if I have a mutation in my skin cell, it's only going to affect me. However, if the mutation occurs in a germ cell, okay, germ cells are sperm or egg or sex cells, they can be passed on to the offspring and be quite harmful. Mutations can be caused by several different factors. Replication errors during replication, maybe DNA polymerase doesn't catch them, and then it goes on copying those errors. Mutagens, agents in the environment that can change the DNA, like UV rays, chemicals, pesticides, and even toxins. So that pretty much does it for 14.4 mutations.